Hello everyone. Today let us discuss about TCA cycle and its regulation. First, let us come to introduction. The citric acid cycle, CAC, also known as the tricarboxylic acid, that is TCA cycle, or the Krebs cycle, is a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates, fats and proteins into carbon dioxide and chemical energy in the form of adenosine triphosphate, that is ATP. In carbohydrate metabolism, acetyl-CoA is a connecting link between glycolysis and citric acid cycle. The pyruvate produced by glycolysis is oxidized to yield two carbon fragments in the form of acetyl group of acetyl coenzyme, that is acetyl-CoA. The cycle consumes acetyl-CoA and water, reduces NAD to NADH and produces carbon dioxide as the waste product. The NADH generated by citric acid cycle is fed into the oxidative phosphorylation, that is electron transport pathway, and the last amount of energy released is conserved in the form of ATP. It occurs in the mitochondrial matrix of eukaryotes. Now let us come to production of acetyl-CoA, that is activated acetate. Pyruvate derived from glucose and other sugars by glycolysis is oxidized to acetyl-CoA and carbon dioxide by the pyruvate dehydrogenase, that is PDH complex, a cluster of enzymes, multiple copies of each of three enzymes located in the mitochondria of eukaryotic cells and in the cytosol of bacteria. The overall reaction catalyzed by the pyruvate dehydrogenase complex is an oxidative decarboxylation and irreversible oxidation process in which the carboxyl group is removed from pyruvate as a molecule of carbon dioxide and the remaining two carbons become the acetyl group of acetyl-CoA. The combined dehydrogenation and decarboxylation of pyruvate to the acetyl group of acetyl-CoA requires the sequential action of 3 different enzymes and 5 different coenzymes or prosthetic groups, that is thiamine pyrophosphate TPP, flavin adenine dinucleotide FAD, coenzyme A, nicotinamide adenine dinucleotide NAD, and lipoic. The PDH complex is composed of multiple copies of 3 enzymes: pyruvate dehydrogenase E1 with its bound cofactor TPP, dihydrolipoyl transacetylase E2 with its covalently bound lipoyl group, and dihydrolipoyl dehydrogenase E3 with its cofactor FAD and NAD. Now let us come to reactions of citric acid cycle. Figure reactions of the citric acid cycle. The citric acid cycle has 8 steps. First, formation of citrate. The first reaction of the cycle is the condensation of acetyl-CoA with oxaloacetate to form citrate, catalyzed by citrate synthase. Second, formation of isocitrate via cis-aconitate. The enzyme aconitase, more formally aconitate hydratase, catalyzes the reversible transformation of citrate to isocitrate through the intermediary formation of the tricarboxylic acid cis-aconitate. Third is oxidation of isocitrate to alpha-ketoglutarate and carbon dioxide. In the next step, isocitrate dehydrogenase catalyzes oxidation of isocitrate to form alpha-ketoglutarate. Magnesium ion in the active site interacts with the carbonyl group of the intermediate oxalosuccinate, which is formed transiently but does not leave the binding site until decarboxylation converts it into alpha-ketoglutarate. Magnesium ion also stabilizes the enol formed transiently by decarboxylation. Now the fourth step, oxidation of alpha-ketoglutarate to succinyl-CoA and carbon dioxide. The next step is another oxidative decarboxylation in which alpha-ketoglutarate is converted to succinyl-CoA and carbon dioxide by the action of the alpha-ketoglutarate dehydrogenase complex. NAD serves as electron acceptor and coenzyme A as the carrier of the succinyl group. The energy of the oxidation of alpha-ketoglutarate is conserved in the formation of the thioester bond of succinyl-CoA. Fifth step, that is conversion of succinyl-CoA to succinate. The next step is the conversion of succinyl-CoA to succinate. This reaction is catalyzed by the enzyme succinyl-CoA synthetase or succinate thiokinase. The reaction releases sufficient energy to form ATP in plants or GTP in animals. GTP can form ATP through a coupled reaction. Now the sixth step, that is oxidation of succinate to fumarate. The succinate formed from succinyl-CoA is oxidized to fumarate by the flavoprotein succinate dehydrogenase. In eukaryotes, succinate dehydrogenase is tightly bound to the mitochondrial inner membrane. In bacteria, to the plasma membrane. FADH2, that is reduced flavin adenine dinucleotide, is produced. Now let us come to the seventh step, that is hydration of fumarate to malate. The reversible hydration of fumarate to malate is catalyzed by fumarase, formerly fumarate hydratase. Now the eighth step, it is oxidation of malate to oxaloacetate. In the last reaction of the citric acid cycle, NAD-linked L-malate dehydrogenase catalyzes the oxidation of L-malate to oxaloacetate. Oxaloacetate picks up another molecule of activated acetate to repeat the citric acid cycle all over again. A molecule of pyruvic acid that enters a mitochondrion is completely oxidized to form 3-carbon dioxide in 1 pre-Krebs cycle decarboxylation and 2 Krebs cycle decarboxylation. Pyruvic acid, it reacts with 4 molecules of NAD, 1 FAD, 2 molecules of water, 1 molecule of ADP plus 1 inorganic phosphate to form 3 molecules of carbon dioxide, 4 NADIs plus 4 hydrogen ion plus 1 molecules of FADH2 and 1 ATP. For Ist acetyl-CoA oxidized by the citric acid cycle, the energy gain consists of 3 molecules of NADH, 1 FADH2 and 1 nucleoside triphosphate, either ATP or GTP. At the end of the cycle, a molecule of oxaloacetate is regenerated. The energy yield from the production of 2 molecules of pyruvate from 1 molecule of glucose in glycolysis is 2 ATP and 2 NADS. In oxidative phosphorylation, passes of 2 electrons from NADH to oxygen drives the formation of about 2.5 ATP, and passes of 2 electrons from FADH2 to oxygen yields about 1.5 ATP. This stoichiometry allows us to calculate the overall yield of ATP from the complete oxidation of glucose. When both pyruvate molecules are oxidized to 6 molecules of carbon dioxide via the pyruvate dehydrogenase complex and the citric acid cycle, and the electrons are transferred to oxygen via oxidative phosphorylation, as many as 32 ATP are obtained per glucose. Now let us come to significance of Krebs cycle. The citric acid cycle is an amphibolic pathway, one that serves in both catabolic and anabolic processes. Besides its role in the oxidative catabolism of carbohydrates, fatty acids and amino acids, the cycle provides precursors for many biosynthetic pathways through reactions that serve the same purpose in anaerobic ancestors. Alpha-ketoglutarate and oxaloacetate can, for example, serve as precursors of the amino acids aspartate and glutamate by simple transamination. Through aspartate and glutamate, the carbons of oxaloacetate and alpha-ketoglutarate are then used to build other amino acids as well as purine and pyrimidine nucleotides. Oxaloacetate is converted to glucose in gluconeogenesis. Succinyl-CoA is a central intermediate in the synthesis of the porphyrin ring of heme groups, which serve as oxygen carrier in hemoglobin and myoglobin and electron carrier. Now let us come to the anaplerotic reactions that replenish citric acid cycle intermediates. As intermediates of the citric acid cycle are removed to serve as biosynthetic precursors, they are replenished by anaplerotic reactions. The most important anaplerotic reaction in mammalian liver and kidney is a reversible carboxylation of pyruvate bicarbon dioxide to form oxaloacetate, catalyzed by pyruvate carboxylase. When the citric acid cycle is deficient in oxaloacetate or any other intermediates, pyruvate is carboxylated to produce more oxaloacetate. The enzymatic addition of a carboxyl group to pyruvate requires energy, which is supplied by ATP. PEP carboxykinase also replenishes oxaloacetate by carboxylating phosphoenol pyruvate in the heart and skeletal muscles. PEP carboxylase also carboxylates phosphoenol pyruvates into oxaloacetate in higher plants, yeast, and bacteria. The citric acid cycle intermediate, malate, is also replenished by the malic enzyme from pyruvate in both eukaryotes and prokaryotes. Now let us come to regulation of citric acid cycle. The flow of carbon atoms from pyruvate into and through the citric acid cycle is under tight regulation at two levels. First, the conversion of pyruvate to acetyl-CoA, and second, the entry of acetyl-CoA into the cycle, that is the citrate synthase reaction. The cycle is also regulated at the isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase reactions. First, production of acetyl-CoA by the pyruvate dehydrogenase is regulated by allosteric and covalent mechanisms. The pyruvate dehydrogenase complex, that is the PDase complex, is strongly inhibited by ATP and by acetyl-CoA and NADH, the products of the reaction catalyzed by the complex. The allosteric inhibition of pyruvate oxidation is greatly enhanced when long-chain fatty acids are available. AMP, coenzyme A, and NAD, all of which accumulates when too little acetate flows into the citric acid cycle. They allosterically activate the PDase complex. Phosphorylation of pyruvate dehydrogenase component E1 by a specific kinase switches off the activity of the complex. Deactivation is reversed by the activity of a specific phosphatase. The kinase is allosterically activated by ATP. Thus, when ATP is high, the pyruvate dehydrogenase complex is inactivated by phosphorylation of E1. When ATP declines, kinase activity decreases and phosphatase action removes the phosphoryl group from E1, activating the complex. Second, the citric acid cycle is regulated at its three exergonic steps. The flow of metabolites through the citric acid cycle is under stringent regulation. Three factors govern the rate of flux through the cycle. It is substrate availability, inhibition by accumulating products, and allosteric feedback. Inhibition of the enzymes that catalyzes early steps in the cycle. The primary control points are the exergonic steps catalyzed by the enzymes citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. First, let us come to citrate synthase. Citrate synthase is allosterically inhibited by ATP, NADH, long-chain fatty acyl-CoA, and succinyl-CoA. Second, isocitrate dehydrogenase. Isocitrate dehydrogenase is allosterically stimulated by ATP, which enhances the enzyme affinity for the substrates. It is also activated by calcium ion. In contrast, NADH and ATP inhibits isocitrate dehydrogenase. Alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA and NADH, the products of the reaction that it catalyzes. In addition, alpha-ketoglutarate dehydrogenase is inhibited by high levels of ATP and calcium ion. Now let us come to conclusion. The citric acid cycle, this is Krebs cycle or TCA cycle, is a nearly universal central catabolic pathway in which compounds derived from the breakdown of carbohydrates, fats, and proteins are oxidized to carbon dioxide, with most of the energy of oxidation temporarily held in the electron carriers FADH2 and NADH. During aerobic metabolism, these electrons are transferred to oxygen and the energy of electron flow is trapped as ATP. Under normal conditions, the rates of glycolysis and of the citric acid cycle are integrated so that only as much glucose is metabolized to pyruvate as is needed to supply the citric acid cycle with its fuel. The acetyl groups of acetyl-CoA, pyruvate, lactate, and acetyl-CoA are normally maintained at steady state concentrations. The rate of glycolysis is matched to the rate of the citric acid cycle not only through its inhibition by high level of ATP and NADH, which are common to both the glycolytic and respiratory stages of glucose oxidation, but also by the concentration of citrate.